Welcome to this lecture on principles of ecology. Today, we're going to explore two of the most fundamental concepts in biology: diversity and abundance. If you have ever walked through a forest and felt that it was teeming with life, you were intuitively sensing these concepts. But as scientists, we need to go beyond just a feeling. We need to measure, quantify, and truly understand what makes one ecosystem thrive while another might be on the brink of collapse. By the end of this video, you will know exactly how to calculate the health of a natural community. Let us begin by imagining you are walking into two different forests. Forest A has exactly 100 trees, but 99 of them are pine trees and only one is an oak tree. Now, imagine Forest B. It also has exactly 100 trees, but it is evenly split with 25 pines, 25 oaks, 25 maples, and 25 birches. Even though both forests have the exact same total number of trees, Forest B feels much more mixed, vibrant, and diverse to us. This simple observation is the heart of what ecologists call diversity and abundance. It shows us that just counting the total number of living things does not tell the whole story of an ecosystem.
Now that we have seen the difference between the two forests, let us formally define our first key term. In ecology, abundance refers simply to the total number of individuals of a specific species found in a given area. It is a raw count. If you count 99 pine trees, the abundance of pine trees is 99. However, raw numbers alone can be misleading depending on the size of the habitat. That is why ecologists often look at relative abundance. Relative abundance is the proportion or percentage of one particular species compared to the total number of individuals across all species in that community. It tells us how dominant a specific group is within the larger picture.
Building on the idea of abundance, we arrive at the concept of species diversity. Species diversity is a more complex metric because it actually combines two distinct ideas into one. The first part is species richness, which is simply the raw count of how many different species exist in the community. For example, Forest B had 4 different tree species, so its richness is 4. The second part is species evenness. Species evenness measures how equal the population sizes of those species are. A community where all species have roughly the same number of individuals, like our Forest B, has very high evenness. True ecological diversity requires both high richness and high evenness working together. Understanding these definitions leads us to an important question. Why do we care so much about measuring diversity? The answer comes down to survival and stability. A highly diverse ecosystem is generally much more resilient to environmental changes, diseases, and natural disasters. Think back to our forests. If a specific disease that only targets pine trees sweeps through the area, Forest A is in massive trouble. Because it relies almost entirely on that single pine species, the entire ecosystem could collapse. But in our highly diverse Forest B, even if the pines die, the oaks, maples, and birches will survive. The forest ecosystem continues to function, providing habitat and food for the animals that live there. To make these concepts useful in the real world, ecologists need to translate them into numbers. The very first step in analyzing any community is to calculate the relative abundance of each species. This formula shows us exactly how to do that. The symbol P sub I represents the relative abundance, or proportion, of a specific species. To find it, we take N sub I, which is the raw count of individuals of that specific species, and we divide it by capital N.
Capital N represents the total number of individuals of all species combined in the entire community. Physically, if the number of a specific species increases while the total community size stays the same, its relative abundance goes up, meaning it is becoming more dominant.
Before we move on to more complex math, let us see this relative abundance formula in action with a concrete example. Imagine you are studying a small community garden. You count the insects and find 60 ladybugs, 30 bees, and 10 butterflies. We want to find the relative abundance of each. First, we must find our denominator, capital N, by adding all the insects together. 60 plus 30 plus 10 gives us a total community size of 100. Now, we simply divide each species count by this total. For ladybugs, 60 divided by 100 gives 0.60. We do the same for bees to get 0.30, and for butterflies to get 0.10. These proportions tell us exactly how the population is distributed. Now that we have the relative abundance for every species, we can calculate the overall diversity of the community. The most common way to do this is by using the Shannon-Weiner Diversity Index. This famous equation accounts for both species richness and species evenness in a single mathematical score. Let us break down the symbols. H prime represents the final diversity index score. The large sigma symbol means we must add up the values for every single species, from the first one up to S, which is the total number of species. For each species, we take its relative abundance, P sub I, and multiply it by the natural logarithm of that same proportion. Finally, because the natural log of a fraction is always negative, we place a negative sign at the very front of the equation to ensure our final diversity score is a positive number. Let us apply this formula to our garden example to see exactly how the math works. We already calculated our relative abundances: 0.60 for ladybugs, 0.30 for bees, and 0.10 for butterflies. We substitute these values into the Shannon-Weiner formula. For each insect, we multiply its proportion by its corresponding natural logarithm. We then add those three mathematical results together inside the parentheses. The natural logs give us negative values, which multiply with our positive proportions to create negative terms. When we sum them up, we get negative 0.898. Finally, we apply the negative sign at the very front of the equation. This gives us a final Shannon-Weiner diversity index of 0.898. This number serves as a baseline measurement for our specific garden community. This baseline score we just calculated leads us to an important question. How does population balance affect this score? To find out, imagine a second garden located right next door to the first one. This garden also has exactly 100 insects in total, and it also has exactly 3 species: ladybugs, bees, and butterflies. So the species richness and total abundance are identical to the first garden. However, this time you count 33 ladybugs, 33 bees, and 34 butterflies. Because these raw numbers are almost perfectly equal, this community has extremely high species evenness. This is a stark contrast to the first garden, where the ladybugs heavily dominated the population. Let us see how this high evenness changes our mathematical diversity score. We will now calculate the Shannon-Weiner index for this second highly even garden. First, we divide each count by the total of 100 to get our new relative abundances: 0.33, 0.33, and 0.34. We set up the equation just like we did in the previous example, multiplying each new relative abundance by its corresponding natural logarithm. When we do the math, we find that the terms inside the parentheses sum to negative 1.099. Applying the negative sign at the front gives us a final diversity index of 1.099. Notice that this score is significantly higher than the first garden score of 0.898. The structure of the formula naturally rewards communities where the proportions are spread out evenly. To truly cement this concept, let us look at a visual representation of what we just calculated. On the screen, you see two bar charts comparing ecosystem A on the left with ecosystem B on the right. The vertical axis represents the relative abundance, or percentage, of the population. The horizontal axis shows the three different species. In ecosystem A, which represents our first garden, you can clearly see a massive imbalance. The first bar towers over the others, showing low evenness. In ecosystem B, representing our second garden, the bars are almost perfectly level. This diagram visually proves what our math just showed us. Even though both ecosystems have three species, ecosystem B is far more balanced. This balance is what makes an ecosystem ecologically stable and resilient to environmental stress. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that abundance simply counts the individuals, while diversity reveals the delicate balance of the entire community. We discovered that true diversity relies on both species richness, which is the number of species, and species evenness, which is how equally they are distributed. By using mathematical tools like the Shannon-Wiener index, we can translate complex natural communities into clear, comparable numbers. We proved mathematically that a balanced ecosystem scores higher, and we learned that this balance is what protects nature from collapsing under stress. You now have the fundamental tools to monitor ecosystem health and understand the true story of our natural world.